So as mentioned in the earlier session, today we will talk about the process of formation of urea. Or the first thing that we must understand, amino acids which as I said that are in excess, they have to undergo the process of deamination, wherein that NH2, the amino group from the amino acid, is detached, and mind well, it is detached as ammonia, NH3. Then it can be eliminated in monotelic animals as directly ammonia, or it can be converted into the nearest possible soluble compound, that is urea. Now this conversion of ammonia into urea is a process that's called as a Citrulin cycle or an arginine cycle or an ornithine cycle, or because on the basis of the scientist who invented this, it's called as Krebs-Hanselite cycle. In short, it's the urea cycle. Now remember, this is happening in the liver because, as I mentioned, all the digested absorbed products are first of all brought into liver, and it is in the liver that their further processing is decided and done. So it is in the liver that formation of urea would take place. The process of deamination, as we discussed earlier, it involves removal of the amino, that is NH2 group, from the amino acid and eliminating it in a suitable way. Now this involves an enzyme glutamate dehydrogenase. It acts on the amino acid glutamate or glutamic acid, and it's broken down into alpha ketoglutarate acid or alpha ketoglutarate, and NH2 is eliminated as ammonia, that is NH3. This is how deamination would happen, and the end product of deamination in this case would be ammonia. The enzyme glutamate dehydrogenase is present inside the mitochondria. So the ammonia that's being generated, it is also generated inside the mitochondria, which will undergo rest of the processing as part of the urea cycle, as part of the Citrulline cycle, which again starts inside the mitochondria. So urea cycle begins inside the mitochondria, and the first compound involved in it happens to be ammonia. And can be directly eliminated as ammonia or can be converted into urea. Now, so far as human beings are concerned, ammonia is found to be highly toxic for various tissues, particularly nervous tissue. In nervous tissue, ammonia is found to be highly toxic, and hence it cannot be treated as the end product of deamination. It has to be converted into something else, which happens to be urea in this case. Now, the first step in urea formation, it takes place inside the mitochondria. The carbon dioxide, which is generated as a byproduct of respiratory process, it is made to react with ammonia, that is ammonium ion, NH4 plus. In the presence of an enzyme called carbamyl phosphate synthetase. Carbamyl phosphate synthetase. As I said, this will happen in the mitochondria. The enzyme is and it's converted into carbamyl phosphate. This will happen in the mitochondria. Now, carbonyl phosphate that is produced in the mitochondria, it will enter the cytoplasm, and then it will be made to react with ornithine. In the presence of ornithine transcarbamylase. Transcarbamylase. In presence of this enzyme, it is made to react with carbonyl phosphate and it is made into citrulline. That's why it's called as Citrulline Cycle. Now, citrulline is acted upon by aspartate. It's acted upon by aspartate and it is converted into arginosuccinate. Citrulline is converted into arginosuccinate. Arginosuccinate is acted upon by arginosuccinase. And it is converted into arginine. Now, arginine will be acted upon by arginase. So as to regenerate the ornithine. During this conversion, arginine to ornithine, a water molecule gets involved, which then liberates the urea. Mind you, the simple formula of urea is CO NH2 NH2. So this is the product of the entire cycle. Meanwhile, ornithine is being recycled, so it is also called as ornithine cycle. The first product is citrulline, so it's also called as citrulline cycle. One of the intermediate products happens to be arginine, so it's also called as arginine cycle. Because purpose of the cycle is to generate urea, it's also called as urea cycle. And because it was first of all explained by Krebs and Henseleit, it's called as Krebs and Henseleit cycle. That's how urea can be generated. Now, one thing we must understand, though urea is being generated in the liver, it would be eliminated through kidneys. Even then, there is no direct blood connection between kidneys and liver. Whatever urea is generated in the liver, it's released into the bloodstream. This blood first of all goes into heart, and then later during the abdominal circulation, it will come down towards kidney. To the dorsal aorta, it will come towards kidney, and that is how whatever amount of urea is released into the blood, entire urea is never eliminated. a part of this urea or part of the blood containing urea which comes to kidneys is subjected to the process of filtration, and only that much blood would be made free from urea. Rest of the blood, it's going to contain some urea throughout. So main purpose of the excretory system would be to keep the urea level within tolerable limits. Excess urea in the blood, which is called as uremia, can lead to a number of complications. Majority of these are related to functioning of the nervous system. We will talk about that maybe in later sessions. So how do you like these videos? Must be enjoying them. Keep watching, subscribe to them, and share amongst your friends and family. Thank you.